of all the people who die from liver disease in the world, 60% of them die because they develop liver cancer, and we think we could probably prevent half of that with aspirin. And so now we've started a big trial in that field, which is running in Australia and will run in the United Kingdom, and we're developing a biospecimen repository, so collecting lots of samples from all the participants who will go into that study, which we'll then use to do some discovery science on, to try and identify blood tests and prognostic markers that tell us who will and who won't develop liver cancer well before they ever develop it. My name's John Olynyk. I'm a liver specialist, gastroenterologist, hepatologist. I work at the Fiona Stanley and Fremantle hospitals for my clinical work, and I work at Curtin University for my academic and research interests. Like most journeys, they start a long time ago, and my interests really in medicine and medical research, which is a large part of what I currently do, really started in the latter part of the 1970s, 1980s when I was doing my medical degree, and I wanted to know what being a researcher was really like, and the way to do that was to undertake a year of research. So I did a Bachelor of Medical Science degree and completed that, and that told me that I really had attributes and interests in research and that I would eventually come back to that. And so I went on and completed my medical training, worked as a junior medical doctor, and then serendipitously ended up getting into training as a gastroenterologist. That meant moving interstate, so I left Western Australia in the latter part of the 1980s and went to Flinders University, Flinders Medical Centre. Started work then, happened to come across some clinicians who had an interest in at that stage, what was thought to be a rare and uncommon disorder called hemochromatosis, which is an iron overload disorder which is inherited and very common in Northern Europe, and the further north you go in Europe, the more common it becomes. Because of those opportunities, I started researching again in the field and started publishing manuscripts and getting research funding to do a higher degree. An MD, as it was called at that time, was a medical PhD, so that's what I did. And through those research exposures and then travelling to conferences and presenting my work, I came across people from international spheres, so the United States and the United Kingdom, who heard my work and who I met and who offered me opportunities to travel overseas. And so after I finished my training as a clinician, a gastroenterologist, a liver specialist, I then went to the United Kingdom and worked in Birmingham at the Queen Elizabeth Hospital, which was at that stage Europe's largest transplant unit, and gained a lot of expertise in liver disease and liver transplant. And then went to the United States and ended up in St. Louis at St. Louis University and continued my research into liver disease, the pathology of how the liver injures, more on hemochromatosis and iron overload. And just after I left, the people I was working with happened to team up with a commercial company in California, and I met them just before I left, and they said they were going to hunt for the gene that caused this disease. Well, a couple of years later, they found it and published it, and because I had a good relationship with them, got access to the genetic testing assays that were required at that time to do large population sort of screening to work out how many people had this condition, how many people with the condition developed disease, how many people who don't have the disease also carry the genes, and look at the clinical characterisation. And for that, I had actually returned to Western Australia and conducted this work in the Busselton study, which is a population study that's been going on since the 1960s, and teamed up with some collaborators. And we genetically tested the entire population. We clinically characterised the entire population and published the world's first study in the New England Journal of Medicine characterising how common this genetic anomaly was and how much disease it caused. And even segueing forward to today, what we observed back then has been recapitulated and confirmed and shown on greater scales, even using studies like the UK Biobank study, which is going on in the UK at the moment. So because of that experience, those publications, I managed to attract NH and MRC funding. My throwaway line is, all you have to do is publish your first paper in the New England Journal of Medicine and you're bound to get an NH and MRC grant. That's the National Health and Medical Research Council of Australia. And after that, from 1999 onwards through to today, I've pretty well been funded consistently in one form or other, either by the National Health Medical Research Council or the Medical Research Future Fund, or the Cancer Council, or a range of other organisations such as the National Institutes of Health and various other research funding bodies. That research expertise then gave me sort of a platform on which to engage with industry, and so I started working with some industry in Western Australia, particularly Resonance Health, which is a local company, to help them develop and validate medical resonance imaging technology, which is now available and licensed by the Therapeutic Goods Administration and the FDA in the United States, to measure iron in human bodies on MRI scanners as part of routine sort of technology. And so I've been able to gain expertise in industry, straight laboratory research in the fields of iron metabolism and liver injury, clinical areas, health economics. I've also looked at various screening strategies and the health economic profiles of those for bowel cancer, etc. within Australia, and published in those fields. I've gotten to meet and train lots of young gastroenterologists, hepatologists and scientists who I've acted as a supervisor for their higher degrees. And then more recently I've managed to help set up and participate in some private community-based clinical trial networks that are funded from overseas pharmaceutical companies that are looking to establish efficient, rapid turnover clinical trials in Western Australia. And through those sort of links, I've now been able to develop more experience in the field of commercial clinical trials. And then eventually my research led me to realise that one of the biggest complications in liver disease, which is the main area that I work in and the most patients whom I saw, is actually the development of liver cancer, and it's got a terrible prognosis. Treatment is difficult, it's becoming more and more common, and it now accounts for after lung cancer, the second most common cause of years of life lost in the world. And I thought, gee, instead of chasing this and treating it and diagnosing it, there must be a way of preventing it. And after a couple of years of putting together a team and a grant to do that and developing some collaborations back into the United Kingdom at the University of Birmingham and the Royal Free Hospital in London, we put together a team in Australia, and last year we were successful in getting just over $5 million from the National Health and Medical Research Council to trial a preventative approach, and that is aspirin in people with cirrhosis, to try and prevent them developing liver cancer, because liver cancer kills, of all the people who die from liver disease in the world, 60% of them die because they develop liver cancer, and we think we could probably prevent half of that with aspirin. And so now we've started a big trial in that field, which is running in Australia and will run in the United Kingdom, and we're developing a biospecimen repository, so collecting lots of samples from all the participants who will go into that study, which we'll then use to do some discovery science on, to try and identify blood tests and prognostic markers that tell us who will and who won't develop liver cancer well before they ever develop it. With relevance to hemochromatosis, because it is in fact a very common condition in Australia, and in fact the treatment of this condition, which results in iron overload, is to donate blood. About 5% of all the blood products in Australia come from patients who are affected with hemochromatosis who go to the Lifeblood Australia service to donate blood. So what I've been interested in is, how do we find all the people in the community who've got this disease? How do we avoid over-diagnosing it in people who don't have it? And how do we deal with the, if you like, the lack of accuracy of some of the testing that's done out there? And by that I mean, for example, testing serum iron levels and what we call the ferritin level in the community is commonly done. There's about 11.5 million tests done in Australia every year for that. About 20% of all those tests come up with a high result, above whatever the recommended reference range should be. But only about 5 to 10% of those high results are actually caused by too much iron. The majority of the tests that are high are caused by confounding factors such as being overweight, obese, having inflammation, having a fatty liver. And so I've been doing a lot of work in populations, including from Western Australia, the Busselton population, from Victoria with Monash Medical Centre, Monash University, examining the accuracy and cut-off ranges that we should be applying in clinical care in Australia, so as to avoid clinicians missing cases that they should find, or over-diagnosing and over-investigating cases that they shouldn't be finding. And that's a body of work that I've recently completed and been writing up. And as a spin-off to that, from the work in Melbourne, we discovered that a very common genetic variant related to the hemochromatosis gene, which doesn't actually cause any significant iron overload problems or disease that we thought, increases the risk about fourfold in men over the age of 70 of dementia. And that had never been observed before. And so now we've actually stumbled across a genetic abnormality that doesn't cause a problem for most of your life unless you're a man and you pass the age of 70, at which point it somehow contributes to an increased risk of dementia. And so we're discovering all sorts of new things along the journey that open doors for further investigation, and that will be followed up at some point in time as well. Whether it's by me or others remains to be seen. Passion comes from excitement and interest. In many ways, I liken my interest in research as very much like a farmer and hobby farming. I do this because I like it, I love it, I'm prepared to put the hours in, and the return on investment may not be great, because everyone knows that research funding is very difficult to get, but for the times that we discover something new and for the things that we find that can impact on clinical care and improve outcomes for patients, it makes it worthwhile. And at this point in my career, where I've segued into a more part-time work mode, the thing that I've sustained is my research. My clinical work I've dropped off, and I'm trying to put as much time as possible into educating and teaching young doctors, young clinicians, all the things that I've learnt to date. And my throwaway line usually is, it's taken me 60 years to finally realise I think I understand the area that I'm in, and now my job is to make sure that you don't have to waste 60 years learning what took me 60 years to do, so that you can actually start from an elevated point of knowledge and hopefully go the next phase and develop things further than me. So I'm quite passionate about education, training, high quality healthcare, and returning to the public. And as a result of all that, just in the last year, I've become patron of Hemochromatosis Australia, which is the consumer group representing Australians who are affected by this genetic disorder, hemochromatosis, and I've been an advisor for them for a long time, but they asked me to become patron because of the work I've done with them, and I have a very active interface with consumer groups to try and ensure that they have just as much opportunity to learn from all the things that I've learnt during my lifetime, as do the specialists such as doctors, scientists, researchers. What keeps me busy are the fact that whilst I've been an active clinician, I have 3 kids and 6 grandkids, and the 6 grandkids aged between 4 months and 13 years certainly offer challenges and opportunities. But one of the interesting things is that my oldest granddaughter, one of the first words she learned to spell was hemochromatosis. So I figured, well, at least I can teach young people about the disorders that I've got. And interestingly, even when I'm out there talking to kids, families or whatever, and you mention the word iron or hemochromatosis, somebody usually in a group says, gosh, I've got that, or someone I know's got that. It's just very common, and often then it leads to further conversations about things. So I quite enjoy grandchildren, family. I like walking, I like being out in nature. I used to play a lot of golf and tennis. I keep promising myself I'll get back into it. But every time I think about that, another research project comes along. So I enjoy a diversity of exposure to research industry, hospitals, professional people, people who have a passion for learning. And, I think my vision is I'd like to think that I'll return something to the community that puts it in a better place for me, having had the opportunities that I've had. I come from a rural background, from a migrant family, and I think I've had a very fortunate life.